Hi, my name is Jack. I am one of the researchers at the University of Southampton, and hopefully today I'm going to be explaining a bit about our recent work in trying to map the GIST tumour and how we explore these cells to find some novel targets in the environment that we can target. But yeah, thank you very much for inviting me to talk today. I really appreciate it. It's something we really enjoy doing, and it's lovely to meet you all. So, perfect. So a little bit about myself. I used to work in an aquarium, the Birmingham Sea Life Centre, if you've ever been. And that really was the start of my scientific journey. I really developed a passion for fish. So I went to university and I got a master's in marine biology. I was sequencing the DNA of seawater. But that led to a job in Bristol where I started sequencing some fish. My interest shifted and I was brought back to Southampton, and instead of fish ecology, I started looking at the ecology of tumours. And interestingly, there's quite a lot of parallels between aquatic ecosystems and tumour ecosystems. Subsequently, I completed the PhD and I'm still in the same team, but now working with GISTs. The team I'm involved with is called the Innovation for Translation Research Group, or the ITRG, and we're quite a diverse group of clinicians, scientists, computer scientists and bioinformaticians, and patients as well. And we all work towards this vision, which I'm going to read out. It's constantly committed to improving the outcomes of patients with solid tumours of unmet clinical need by delivering better, less harmful, and more precise treatments. And I've highlighted three words there of the better, less harmful, and more precise. And this really underlines all the work that we're doing as a team to try and develop these new therapeutics to help in these solid cancers. So I work with GISTs. I've listed here three problems. The first problem is there's a limited treatment options. Over 20 years ago, imatinib has kind of revolutionized GIST treatments, dramatically improving patient outcomes. But there's quite often resistance, as you've heard, so there's second line, third line, fourth line treatments. But it's an evolving problem. There's also pause, so there's certain mutational subtypes of GIST that don't respond to these tyrosine kinase inhibitors like imatinib. And lastly, imatinib can be quite unpleasant sometimes. So that really kind of links back to our overall vision of our group of this better, more precise, and less harmful. And developing these new treatments, one of the problems is the complexity of the tumour. I've been talking a lot about ecosystems. If we look at the tumours in ecosystems, it's quite a complicated collection of different kinds of cells, all interacting and communicating with each other. So finding targetable things that we could focus on in the tumours, it can be complex. And lastly, the problem three is the lack of good models. So we're able to grow GISTs in a lab, but whether these cells accurately represent a tumour in the patient is something we're trying to develop. Once we can develop these good models, we can start to look at these interactions and communications between different cells, which we can target, and we can then test novel therapeutics on these models as well. So GISTs, the cancer cells don't exist alone. There's a variety of different cell types that support and communicate with the tumour. It's called the tumour microenvironment. And so if you have the cancers, these grey cells, there's other cells that support the tumour and communicate with the tumour, helping it grow, helping it spread, helping it survive your own body's immune system from killing it, and it can even help the cancer become resistant to treatment. So the two questions we're kind of trying to focus on with this environment is what cells are present in GIST tumours and how do these cells interact with one another. I have this analogy here about how technology is improving. 10 years ago, the only way we could look at a tumour, any tumour, is as if we are looking at a smoothie. A smoothie contains all these different fruits, but they're all blended, and it's hard. You can try and work out what that smoothie consists of, but it can be quite difficult. in 2015, new technology was invented which allowed us to explore each individual cell of a tumour. It's like looking at a fruit salad, so we can see that there's a bit of kiwi or a bit of banana or a bit of strawberry. The way we do that is through microfluidics. So I've got this animation here, and we use the property that oil and water don't mix, similar to how vinegar and oil don't mix in a restaurant where you dip your bread in. And we can create these droplets, these bubbles that you can see being produced on the right. So if we can take a patient sample, we can chop it up until we have single cells, and we can pump it through this microfluidic device. We can then also pump through these barcoded beads, and once we get a single cell and a barcoded bead together in a droplet, the cell is exploded, and the information of what that cell is and what that cell is doing is captured onto that barcoded bead. We can then collect those barcoded beads at the end, and because of that barcode, we can really pinpoint the exact cell that all this information is coming from. And we can produce a map that looks something like this. This is called a U-map, and each single dot on this plot is an individual cell, and its position is based on what that cell is doing. So cells doing a similar sort of thing clustered together. So even that top left corner, there's that blue cluster of cells labeled GIST. Those are the cancer cells. But you can see that there's quite a diverse range of different cell types that are there, including ICCs, which stand for interstitial cells of Kajal, which are the potential cell of origin of GISTs. This was the first patient that we did this to back in 2018. And we can then start to explore what these clusters of cells are doing. So there's this type of cell here called a fibroblast, and we can look at what the cell's doing, and you can see that it's communicating with the GIST cells, promoting their growth and supporting the spread of this cancer around the body. Similarly, again, there's these immune cells, and we can see here that these cells are promoting the growth again, but also suppressing the immune response. So it's stopping your own immune cells from attacking and killing the cancer. Over the past few years, so that was in 2018, we've added to this data set and combined it with other data sets from around the world from different labs. And we can really start to build this cell atlas. And so we're hoping to generate a map of all the different types of cell that we can potentially find in different GISTs. This is a collection of all the different mutational subtypes, including the common KIT ones and different types of patients that have developed resistance as well. And again, we can try and kind of separate, see what cells are there. So on this plot on the right, you can see that the cancer cells form this cluster in the middle in red. You have a large portion of immune cells in green, and again, these supporting stroma cells in blue. So from this, we can then start to explore what these cells are doing, how they're communicating. But one thing this lacks, it doesn't tell us whereabouts in the tumour this is from. I think we heard a little bit earlier that GISTs can grow quite large, and they're quite heterogeneous, so different areas of the tumour are different. And all the spatial information is lost when we do this technique. But technology has progressed. In the last couple of years, new technology has been discovered or invented that allow us to retain that information about whereabouts in the tumour these cells are present. I've added to this fruit salad analogy and included a fruit platter. And so now we can see that there's a line of pineapple next to a line of melon. And we can apply this to the GIST tumour samples that we obtain. So how it works is we get a 2D cross-section of a tumour. It looks something like this on the right, and we can start to see some of the heterogeneity across the tumour. We can see blood vessels and some large architectural subtypes. Oops. I've gone a bit too fast. Oh, no. And we can, similar to the barcoded beads you saw earlier in creating droplets, but instead of introducing them into droplets, we can apply this slice of tumour on top of a slide containing barcoded spots. And again, you can see on this right, there's a variety of different colours, which represent there's different types of cell present. The real power of this technology comes when we combine it with the cell atlas that we've generated. And we can start to see the cell types that are present, see how they're interacting, and see the large changes across spatially in the tumour. So we can see that there's lots of immune cells that are present in the GIST. These are the cells that you should expect to be attacking the cancer cells, and why they're not is something we're trying to explore. We can try and apply drugs to this to try and encourage the GIST cells to communicate with these cells, and so they start acting the way they should. There's a new starter starting in the next week that's looking at introducing a drug called an EZH2 inhibitor, and we're hoping that's going to cause the GIST cells to develop this communication with this immune cells that are present throughout the tumour. There's also this area in this top left, which is labelled GIST cycling. These cells are cancer cells, but they're rapidly dividing, and why this area is more rapidly dividing than the others is something that we're currently exploring. One limitation that we have is that each of these dots is not a single cell. Each dot is about 5 micrometers across, and your average cell is about 10 micrometers. And so it's probably about 5 cells per spot. And so some of this resolution we lose with this technology, but again, technology advances. And in the past month, we've started using this new platform called Cosmex. And this allows us to get this single cell resolution, but also spatially as well. So this is another plot similar to the last one, but instead of having this individual spot, which counts as an area, each spot here is a single cell. And again, we can start to look at the different cell types, the diversity of the tumour, and see what it is targetable. So onto that last problem, there's a lack of models that we can utilise to explore the communications between different cells in the tumour, as well as testing new therapeutics. We are co-led by a surgeon who routinely resects GIST from patients. And so we're able to get these fresh samples straight from theatre. We're also able to get blood. And from these, we're able to derive a variety of different cell types that you would find in a GIST. And then we can try and recombine them, and this is hoping to develop a more patient-accurate representation in the lab. From this, we can then explore these communications and test new therapeutics. We can do this quite successfully, so we're routinely deriving GIST cells from patients that come through Southampton. We can stain them with different colours. And we use a marker with a fluorescent green tag, which just confirms that the cells that we have are GIST. And you can see that there's some green showing, so these are GIST cells. We're also able to get immune cells, and also a type of cell called a fibroblast. And these are the supporting cells that encourage the tumour to grow, as well as encourages the tumour to spread around the body. So the next steps is to recombine these different components of a tumour in the lab. This is some of the work in our team that's been done on esophageal cancer, where we've combined the cancer cells along with this type of cell called a fibroblast. In this animation on the right, the cancer cells are in purple or red, and the fibroblasts are in green. And you can see how the fibroblasts embed themselves within the tumour. One thing that we've been looking in addition to these models is to use a PDE5 inhibitor to try and change these fibroblasts to, instead of helping the cancer grow and spread, to actually inhibit the cancer from growing. So in summary, what I've been focusing on with GIST in the lab is to explore these different types of cells, this environment of a tumour, see how these cells communicate with each other, see how these supporting cells promote the growth and how they resist treatment. And then if we can reprogram these supporting cells to actually fight the cancer instead of helping it. I've got a photo here of a lot of different faces that have all been involved with our GIST work. But I just want to highlight these 4 people in the square boxes in the top right. These are the patients and parents or carers of patients with sarcomas or other cancers. And they really help us guide our work that we do in our team and really help our vision as we grow and explore different avenues for treatment. And we're continuously looking for more patients to be involved with our research. And so if anyone's interested, feel free to come talk to me and Zoe. I think that's me done. Yeah. Thanks.